Welcome to the second topic in Chapter 15, Protein Sorting. Here are the objectives for this part of the chapter. We can't talk about organelles without talking about the proteins that make them special. Each organelle in our cells has a unique set of proteins on its membrane. So let's talk a little bit about how proteins are sorted into the organelles that we know. In this section, we are going to talk about how proteins are imported into the organelles. We're going to talk about how signal sequences can direct protein distribution. Next, we are going to talk about how proteins are imported into the nucleus, the mitochondria, the chloroplast, and the ER. And lastly, we are going to talk about transmembrane proteins and how they get to the plasma membrane. As we just mentioned when we talked about the ER, proteins destined for the membrane are produced in the endoplasmic reticulum. Proteins whose role requires them to be on the internal side of an organelle are produced within the cytosol on free ribosomes. How then is that protein, which is produced in the cytosol, sorted into the organelle it was made for? There are three main processes. The first of these is transport into the nucleus. This occurs through importation through nuclear pores. The second way is transferring across membranes. This happens with chloroplast, mitochondria, and the ER. This process is completed with the aid of protein translocators, which are located in the membranes. The last way the proteins are passed to other organelles is through vesicle transport via the ER. So those are the ways that the proteins get into the organelles, but how are these proteins directed to these organelles? This happens through signal sequences. What is a signal sequence? This is a stretch of amino acids, generally 15 to 60 amino acids long. Once these proteins have been moved to the appropriate organelle, the sequence is usually removed from the protein. So now we know how proteins get to where they need to be. Let's talk about how they get into the organelles. We are going to start with nuclear pores, but we'll then talk about importation into other organelles. As we know, the nuclear envelope is a double membrane with pores scattered throughout its surface. We've talked about these pores when we discussed how the mRNA exits the nucleus, but let's investigate how stuff gets into the nucleus. This pore is a large protein complex consisting of about 30 different proteins. Water and other small molecules can easily pass through this pore, but how do larger molecules get through the pore? It starts with a nuclear localization signal. This is an example of the signals we just talked about. When this signal is detected, the nuclear transport receptors will bind to this region of the protein. This receptor then directs the transport of these proteins through the nuclear pore by interacting with these scary-looking fibrils that surround the pore. Once the protein is past the fibrils, the receptor will then work to guide the protein through the tangle of nuclear pore proteins. Think of this almost as a jungle guide helping someone through a deep jungle. As you would imagine, this process requires energy input to help cross the membrane. This image demonstrates how GTP hydrolysis is used to help import nuclear-bound proteins into the nucleus. Now let's look how proteins are imported into mitochondria and chloroplasts. Because they also have double membranes, the import into these organelles differs from other organelles. The protein in this case is completely unfolded. The signal sequence binds to the receptor protein. This protein will then move across the membrane to the contact site. At the contact site, the protein is moved across both membranes simultaneously through the protein translocator. This will then move the protein into the matrix or the stroma, where it is refolded and the signal sequence is removed to be recycled. What's weird about the ER is that we don't think about it needing proteins imported into it. The rough ER makes protein, so how could it possibly need proteins? Well, it is through the importation process that the proteins made in the ER are made. The mRNA is threaded into these bound ribosomes, and the protein is made as it enters into the ER. These bound ribosomes are identical to the free ribosomes that we discussed when we talked about protein production. The only difference is that these ribosomes are attached to the cytosolic edge of the ER. These can sometimes create the polyribosome we discussed in Chapter 7. So how does the ribosome know if it should attach to the ER or not? It all comes back to the signal sequence in the amino acids. That will direct the ribosome to attach itself to the ER. Once the protein is in the ER has to distinguish the function of the protein. The transmembrane proteins are those that are destined to be embedded in the plasma membrane or the membrane of other organelles. There are also proteins that will remain free of the membrane. Let's look at both of these proteins, starting with free proteins. Free proteins are always water-soluble and enter the ER just like we discussed in the previous slide. However, we have a few differences. The first is this signal recognition particle, or SRP. The job of the SRP is to recognize the signal sequence that indicates that the protein is destined for the ER. It then binds to the protein and the ribosome and guides it to another protein as the SRP receptor. This receptor is embedded in the membrane. The binding of the SRP to the receptor helps the ribosome and the translocation channel associate. The ribosome may then finish the protein synthesis. At a variable point during synthesis, the protein signal sequence is removed from the protein by signal peptidase. After the entire protein has entered the lumen, the protein is folded and released. While it is fairly simple to release the free proteins into the lumen, the importation of transmembrane proteins is slightly more complicated. I want you to pay close attention to this slide. It will be crucial when we talk about cell communication in the next chapter. There are two ways of creating transmembrane proteins. The simple way is for a protein that passes through the membrane only once. And the more complicated way is where there are multiple pass-throughs. Before we get too far into the mechanisms, let me remind you for a second of the sidedness we talked about in the membranes. The part of the protein that ends up on the lumen side of the ER is transported to the plasma membrane, where it will be the extracellular portion, while the portion that remains in the cytosol when embedded in the ER will remain on the cytosolic side once shipped to the plasma membrane. So let's start with the simple pass-through mechanism. As you can see, the signal sequence is at the N-terminus of the protein. The protein then enters the translocation channel, and the protein is passed through the channel until the stop transfer sequence is reached. This causes the pore to release the protein into the membrane. When this happens, the protein becomes embedded in the membrane and the signal sequence is removed. The protein is then ready for transport to its final destination. In the more complicated model, the signal sequence is not at the end of the protein sequence as it was for the transmembrane protein that passes through the membrane only once. Its location causes the protein to fold like a hairpin to enter the channel, and the process proceeds normally until the stop transfer sequence is reached. This forces the channel to spit out the protein into the membrane again. However, the protein spans the membrane twice, so the signal sequence cannot be removed from the protein and remains with it. Some proteins span the membrane numerous times, and we believe this embedding comes from multiple pairs of signal sequences and stop transfer sequences. Remember how this happens. This is an important process for creating communication proteins, such as the G protein.